Welcome to The Long Run. This is a podcast for biotech adventurers. I'm your host, Luke Timmerman. Today's guest is Valerie Daggett. Valerie is the founder and CEO of Seattle-based AltPep. This company is working on an unusual diagnostic and therapeutic strategy against Alzheimer's disease. I wrote about the company in June 2023 when it raised $53 million in a Series B venture financing that included Section 32, Alexandria Real Estate Equities, and Eli Lilly, among others. At the time, I wrote, quote, "What if you could screen everyone at age 40 with a simple blood test that could detect trace amounts of the toxic forms of amyloid beta protein that build up over 10 to 20 years in people with Alzheimer's? What if the blood test had 99% sensitivity and specificity to the toxic forms of the protein structure, ignoring normal forms? What if you could go a step further after a positive early diagnosis and give a treatment that binds the same way but is optimized to clear out those misfolded proteins before they start bunching up into plaques that damage neurons to the point of no return?" This might sound like daydreaming in Alzheimer's diagnosis and treatment, but Seattle-based AltPep is imagining such things and has secured $53 million in a Series B financing to pursue this plan. End quote. Now, Valerie, in addition to her day job at AltPep, is still a professor of bioengineering at the University of Washington. She has been on the faculty there for 30 years. Her academic research has been building and building over time, pointing toward this moment in which her team can make peptides that are so exquisitely able to distinguish toxic forms of amyloid beta protein from the garden variety. They can also capture and remove those proteins. In this conversation, Valerie describes how her research evolved in this direction, why she decided to continue working on it in the business world to see if it can fulfill its potential, and where she thinks it could go. Please join me and Valerie Daggett on The Long Run. Valerie Daggett, welcome to The Long Run.
Thank you.
So I have to say, Valerie, I'm really pleased to be at your company in person here in Seattle. There's something about just seeing people where they work, being in a lab, especially in this new facility where you have beautiful views of Lake Union and the Space Needle, and you're right in the middle of a high science neighborhood. So it's great to be here.
I agree. It's fantastic to be here, to have these fantastic labs and to be in this this hub of what's going on in biotech in Seattle.
Awesome. Well, we're going to get into what you're doing here at AltPep in a bit, but I like to start with a little bit about you. So where are you from originally?
Portland, Oregon. And then I went to Reed College and then worked for a couple years in the lab and became enamored early on with computational studies.
Okay, well, let's back up. We'll get there in a second.
Okay.
But Portland, Oregon, what kind of schools did you attend?
Public schools early on, and then neighborhood schools, that sort of thing.
So how did you get the bug for science?
Not from my family. I'm not really sure. It started with a love for math. And so I was accelerated in math and had, in fact, my last 3 years of high school, one-on-one teaching of doing proofs and things. And that excited me until I saw it applied to physics. And then when I saw it applied to chemistry and then applied to proteins, and that's where I really was like, okay, this is it. This is what I want to do.
Wow. Now, what years are we talking about? This been high school?
Oh, geez, that you really, that's not fair. So I graduated in 79.
Okay. So you got the interest in chemistry, proteins very early on. Now, what
proteins came in college?
Okay. So that came later, but it starts with math.
Yes.
So you had a gift and someone recognized it.
Yes.
And decided to get you some special tutoring.
Well, they did it through the school, in fact. There were teachers that said, okay, we'll teach, these classes and special classes.
Mm-hmm. And And Reed College, for those who don't know, famous for, well, lots of things. Kind of a quirky curriculum, private liberal arts, academically challenging, where Steve Jobs went
Yes.
to school.
Yes.
Roger Perlmutter, too, by the way,
Yes.
biotech. Did you know any of these people there, by the way?
No, they were ahead of me.
Okay. Okay. So what did you study at Reed, and what captivated your imagination in those days?
It was the same theme. I started by with math, thinking that would be it. And of course, it's a liberal arts education, so you're taking all kinds of other courses as well. And then moved to physics, and then at that point moved to chemistry. And my degree was in chemistry, and that's where I was exposed to proteins, and really became enamored with trying to understand how proteins fold and unfold, and what they do.
So it starts with classic chemistry, organic chemistry, and then veered into biochemistry.
There was certainly physical chemistry as well, and that's still a theme in what we do now. So biophysics, physical chemistry.
So what were you thinking you were going to do when you grow up back then?
So Reed had a very good chemistry program, and it's also a place where they indoctrinate people into going into academia. So I was going to be a professor. I was going to go into academia, but I wanted it to be my choice, not, be kind of being pushed into it. So I took a couple of years off. I traveled a lot, and I worked in a lab at Rutgers to see, hands-on what it would be like. Now, Reed, you have to do a senior thesis. And so I had, dipped my toes into this, but then wanted to really make sure it was the right path. And I loved it. And so then it was on to grad school and postdoc and that sort of thing.
The lab work at Rutgers was this interim step that got you interested in, yes, you do want to pursue the PhD and go the academic route?
Confirmed it. Yeah.
So where did you go to get your PhD?
UCSF.
And what brought you there?
A variety of things. I was considering many places and kind of going back and forth on some. And I really liked what they were doing at UCSF. There were a lot of exciting things going on then. And it was, they were bringing together computational and protein work, kind of like no other university. And so that's what really grabbed me. And then San Francisco. I mean, I loved San Francisco.
And this was early days of computers. This is the 80s.
Yes.
So what were you thinking that would help you and your colleagues do at the time?
Well, I mean, when you're young, maybe you're thinking about that or you talk about it, but you're really just immersed. It's pleasing you, right? It's something you do because you love. And so then it isn't, I think, until I got older that you start to feel more of a duty or what can you do with this? But and especially being indoctrinated into pure research, and this is the right way to go. It was driven by the research, the questions, the answering the question. What's the deeper question?
I think you kind of answered this, but what was it that you loved about biochemistry, structural biology as you went through graduate school?
I like to see things. I like to see, and not just it. I found with experimental work, and this came out at Rutgers, is that you would see something, but it was so phenomenological. Something would happen. You'd start with this, you'd end with this, and you couldn't see what happened in between. And I really wanted to know what was going on in between. And I had already started studying protein folding at Reed, doing experimental studies, and it just felt limiting. And so it kind of went back to, well, I want to bring in the math and the physics and such, and you can't do this experimentally, really map those processes, is trying to do it computationally. And there were a couple of great labs at UCSF doing that.
And this was, well, then and until very recently, one of the very big problems in biology was how do you predict or determine the folding structure of the protein based on the underlying amino acid? People did not know this.
Right.
How that happened or how to predict it.
Well, the predictions you're seeing don't tell you how it happens. It's the same problem I was talking about. So that doesn't solve the problem. I wanted to know how it happens. We were the first to do that and develop methods to simulate this process realistically so we could map the pathway. Prediction only says, okay, I predict this to be the structure. It doesn't tell you how you get there. Now, the reason you want to know how you get there is that there are so many diseases that are not necessarily due to folding how you get there because, the body finds a way or it doesn't, and there's chaperones and such. But proteins will fold correctly, then they unfold. And that's with single nucleotide polymorphisms, the amyloid diseases we study, all kinds of diseases are associated with that. And that's what really became of interest to me.
So you dug in on this protein folding, understanding that process.
Correct.
And this is the kind of thing you can spend an entire academic career on.
Yeah, I have.
and you did. So I think you did a postdoc stint at Stanford for a while.
I did. I was a postdoc with Michael Levitt, who we were working on these methods for simulating the proteins and doing the first studies of protein unfolding. And he got the Nobel Prize in chemistry in this area. Was kind of the grandfather of the computational studies.
Yeah. Yeah. So, and during these years, of course, computation was getting better, faster, cheaper, and presumably a little bit more useful in helping you map out some of these steps on the process.
Absolutely. Yes.
So what brought you to the University of Washington? Gosh, almost about 30 years ago.
Yes. It probably was triggered somewhat by, it's not science, but lived in San Francisco. I loved San Francisco. Moved the final year to Menlo Park by Palo Alto, and then was seeing the Bay Area as this expensive place and very crowded. And I'm a skier, as you heard, and you would go up and there was just so much time in the car to try to go skiing. You couldn't, and then, or if you're going to Yosemite or something, you get out and all of a sudden there's a bus full of tourists, and you couldn't get away from people almost. And so I started to get a yearning. I never thought I'd want to come back to the rain, but the Pacific Northwest.
Yeah.
Wanted to move back. And I wasn't really looking for a job yet. I just kind of dipped my toes into it and to see what was available, and then got one, moved here, and I've been here ever since.
But then and now, the University of Washington was a powerhouse. It's much bigger now, but what kind of environment did you find yourself a part of as a early faculty member?
Well, this is one thing I really like about UW, and something that hit me when I first came to interview is the culture, the people, the collegial nature of things. And it was so nice when you're starting out, it's kind of tough. You have all these great things you want to do, and you get a startup package and that sort of thing. But you can't really do a lot of these things on your own. And at UW, I just found so many people were so generous with their time, their expertise, working together. It's just very, very collaborative. Reminded me of UCSF. UCSF was like that. And so that's kind of critical. And then, I have my own sort of theory for this, which is probably wrong, but they were... Back when I was moving up here, some were saying, you're committing professional suicide going up there,
The hinterlands.
Yeah, exactly. But I think that also helped to create this culture we have here. And so people would rely on each other. And it's not that you don't collaborate with others and that sort of thing up far, but you look inward. You aren't just always the Bay Area. Sometimes you're in competition with the person right there. So it's a little tougher. But one thing I really like, so I see that in academia at UW, but then it feels like it carries over to the private sector. It just, I love the interactions with other biotech founders and people here.
So this is the early 90s. Did you join, which department was it? Was it bioengineering at the time?
No, at the time it was medicinal chemistry.
Okay. small molecule or large molecule?
No, I was setting up doing protein studies. That's where we started, in fact, the first simulations of amyloid proteins. And I was collaborating with Stan Prusner at UCSF still. And so we would do experimental work. We would do the computational work and then work with experimentalists. And that's been the story throughout my career as a computational person. But medicinal chemistry departments and pharmacy schools were more receptive to computational work early on. And in part, getting back to what you asked, because of small molecule work and drug design. And so we didn't fit that per se, although I can do it. I have done it. But they were, very receptive to, okay, let's go to bigger molecules. Let's try to understand this and relate it to disease processes. And some of the other departments like biochemistry, bioengineering didn't even really exist at that time at UW, but it was just beginning. But some of them were just, they were more focused on the experimental side, and computational work hadn't really come of age yet.
You mentioned Stan Prusner. He was at UCSF.
Yes.
And he won the Nobel Prize for, I think it was the discovery of the infectious prions, the mad cow disease.
Correct.
And here's an example of a misfolded protein that causes a severe pathology we had no knowledge of. Wow, that was an interesting discovery and paved the way, or stirred, imaginations about what other things we might learn.
Oh, absolutely.
About misfolded proteins.
Yes.
Can you sort of alluded to this, but can you talk about like what you thought you wanted to do in terms of setting your research agenda long term when you got started? What were the big questions that animated you as a beginning faculty member?
So I'd already started this work at Stanford and doing the unfolding, but it was just a couple of simulations, and you're making predictions, and you have nothing to back it up basically. But journals are accepting it; they were receptive. But it became clear we had to really get validation through experimental work. I started a long time collaboration with Sir Alan Furst at Cambridge. And that was critical because he was also at the MRC and had facilities to do essentially anything. And so you can have these dreams of doing things, but you got to do the steps in between to show it works and to validate it and, make sure things are robust. And so we began, we still collaborate almost to this day. And he, so we would make predictions. We looked at a number of different protein systems and then do the experimental studies, sometimes with my people going over there to do this with his. And so what we did is establish the foundations that the methods work, that we can do unfolding, that we can bring a refold from there, and just all the things you need to do from a computational point of view. And then validated these things experimentally. And about the same time, when I started my lab here, my first grant was with Stan Prusner, in fact. So, and then we were working on A beta, which is how I got involved with his group, and then the prion protein. So this was going on, but we didn't have the validation there it's not firm enough, right? You don't have the structural information and things like that. And so they were going on in tandem, but always with an eye to the disease. And then once we validated the methods, it was much easier to make the case for the amyloid proteins and what we were doing there.
Now, was the amyloid beta hypothesis as the one of the principal drivers of Alzheimer's, was that out there, widely known or believed in by many scientists at the time?
It was more nebulous, for sure, but it was out there.
Mm-hmm. Mm-hmm. Did that strike you and your collaborators as an important area of investigation to follow through on and see, gosh, because if it is a principal driver, one of, or maybe the one, that would be really important for Alzheimer's.
Oh, absolutely. We jumped right in and started doing simulations of A beta, building plaque models, all kinds of things. Yes.
And you really were setting up the lab so that you had both computational and experimental capabilities in place.
Outsourced experimental capabilities at that point.
Oh, okay. So the wet lab work was being done somewhere else.
Right. And so that was done often in collaboration with people at UW. A large part was with Alan First, as I mentioned, and such, but it was all outsourced until I moved to the Department of Bioengineering.
Okay. So you brought these things back together, but there was a recognition early on that these two things couldn't exist in a vacuum. They needed to work together in some kind of hand in glove fashion.
Absolutely. And so also at the time with protein folding, as it was really being fleshed out, that became, both sides started to accept that, right? So the experimental and the computational, that you can't stand alone and that you really need each other. So experiment, it's sort of, you've got one thing, you start with something, you jump to something else, you want to know what goes on between, you need the computational work as a framework for that. And the computational work can't stand alone. You need to, assess and validate.
Mm-hmm. Now, okay, so let's fast forward a little bit. Years go by, computation gets better, faster, cheaper. The knowledge base about proteins and folding and their role in disease pathology just keeps getting bigger and bigger and deeper.
Yep.
Was there a point when, what point where you felt like, wow, we are really entering like a new era where we can really bring these tools to bear in a faster and more predictable fashion to someday help people?
So that happened really maybe 15 years ago or so. So we started expanding what we were doing on the protein folding side, just for a second, not the amyloid side, to explain this. So we started saying, how representative is anyone's system anybody looks at? Because experimentalists pick systems where they can get answers easily or produce the protein easily, that kind of thing. And so we started an effort we called dynamiomics, and we did simulations of representatives of all known protein folds. It was the biggest collection of structures in the world in simulations. And so we did native proteins, we did unfolding and things like that. Now, what that bought us is, and then we did a stint in developing databases. We were supported by Microsoft. We did big data analyses, developed methods, all kinds of things. Now, that is still in that category of kind of the validation, what can you really get out of simulations and trust and that kind of thing. But it provided a really great set to compare against the amyloid simulations. And so what we found is that we started looking at more and more amyloid proteins, moving beyond A beta and the prion protein. And there are over 50 diseases caused by different proteins and peptides that undergo this unfolding, refolding, misfolding sort of process. And so we started looking at more and more of them and found that they were funneling through a new structure. So we discovered a new secondary structure, which we call alpha sheet. Now, that right there, for a long time, we thought, okay, this is an artifact. So that led to a couple of big changes. So we're investigating that. But part of the way to check that was against the dynamiomics study, because there we had thousands of simulations that don't show that structure, normal proteins that don't show that, whereas amyloidogenic proteins were forming that structure.
Can you pause just a little bit what you called alpha sheet?
Yes.
What is this? It's some kind of marker on the protein that is only on the toxic forms and not the garden variety vanilla A beta that
correct.
Might be elsewhere.
Correct. So the alpha sheet, when we first proposed it, we called it gamma sheet, which I wish we had stuck with, alpha, beta, gamma, because then it gets confused with alpha helix as alpha sheet. But there's a reason for that because of the conformation at the local level. But alpha sheet is essentially, if you think of beta sheet, which is the common structure and the structure in fibrils and plaques, is a crankshaft motion, every other amino acid. And what that does at the atomic level of the main chain is it creates a molecular dipole so that you have all your carbonyl oxygens lined up and all your NH, your positively charged amide hydrogens off your nitrogens lined up. Now, this imparts special properties to the peptide. So you can have a peptide that has that structure, and for instance, it inhibits aggregation in amyloid systems. You take that same sequence where it doesn't have that crankshaft motion, where you don't have that structure, and it doesn't work. And it has a completely different structure. It's not alpha sheet.
So here's something where the structural biology is telling you something
Absolutely.
that you really could not infer straight up from a gene mutation or some underlying amino acid abnormality. That's not going to point you in this direction. But you found something here. And furthermore, you were able to design, the next step would have been peptides,
Right.
a specific targeting agent that could bind with this this crankshaft, as you call it,
Right.
that leads to the toxic form.
Yes, absolutely. And so that's what we began to do. It said, how can we recapitulate the structure we're seeing in the early stages of amyloidogenesis in multiple simulations of protein, amyloid proteins? And then they have different sequences, they have different structures, initial structures, they have different functions, and yet they're funneling through this structure in the computer. So then we designed compounds that would have that structure. We did this in silico. We designed thousands in the end. We ranked them computationally again in silico, and then we began making them synthetically. So we designed a template so that we could stabilize that crankshaft structure and made those synthetic peptides.
Now, were you thinking that this might someday be the basis for a therapeutic?
Yeah. Yeah, I wasn't necessarily, crying it from the rooftops or something because it was so early, but yes, that was the hope. So the idea was, and we made that hypothesis, and we got early on PNAS to publish it before there were any experimental data to back it up. But we said, look at these proteins are funneling through this structure. This is a unique non-native structure. We hypothesize it's associated with toxicity in these early oligomers, these small aggregates that form. And as such, if we can design something that'll be complementary to that and neutralize it, then we could have an inhibitor of this.
So when did this mature to the point where you thought maybe this could become a company?
Well, there were some steps in between. So I was still, when this was happening, making the discovery and such, and when we published a number of papers on this, we were connecting up as we could with experiment, but it was limiting. So at the point where we were making designing the compounds, we needed a lab to do it in. And at that point, I made a lateral move as a full professor to the Department of Bioengineering. And that was critical. The company wouldn't have happened without that. And so what we did is had a wet lab, now still relied on using, some equipment here and there, again, back to the collegial nature of UW, but we were able to make our own peptides, make libraries, check them, and really check this hypothesis. And then by the compounds are important, not just as therapeutics or for diagnostics. They were also important early on because you have to show it's real. It's not a computer artifact. You have to show that you've got that structure, which took many years and a lot of work because peptides are notoriously kind of conformationally ambivalent. And so, but we did get good NMR structures. We determined the spectroscopic signatures of alpha sheet. And that's critical because you can't go into the amyloid systems if you don't know what you're looking for. And so whenever you do spectroscopy, these biophysical methods, you're comparing against what's been done in the past with model compounds. So our first compounds, in essence, were the model compounds that allowed us to then go into the A beta system, for example, isolate the conformational oligomers, the toxic oligomers in different states, and then look for their structures and say, do you have this alpha sheet structure or do you have the beta sheet signature? That sort of thing. And so at that point, we were able to engineer the process so that we could, for the first time, really get in there and characterize what was going on structurally and with the aggregation that also occurs as a part of the process of amyloidogenesis, and to do so with good biophysical metrics for this. And that took moving to a new department and being able to do the work in-house.
So this is all happening at the university. You are developing the molecules. You're seeing experimentally that they are binding the way they're supposed to with the right properties. In parallel, meanwhile, the whole pharmaceutical industry has put a lot of money and effort into the amyloid hypothesis, developing antibodies and other things to work on clearing these toxic plaques that accumulate with time, and a big graveyard of failures occurred there. When you saw all that happening, were you disheartened at any point, or did you think, no, we actually have a different, a very an orthogonal approach, binds differently, works differently, let's keep going?
The latter. So to kind of go back to this idea of how this structure is changing and the aggregates that are forming, the field was focused on plaques and also taking out the starting structure, BACE inhibitors, and binding it or affecting, its production, A beta 42's production. So we then were saying, okay, no, there's something earlier that's going on. And there were other people in the field saying that. And there was more and more coming out about oligomers, but nobody had a handle on the structure and whatnot. That came by an orthogonal approach, and you couldn't predict it. Machine learning won't give you something novel like that. You have to feed it something to give you these answers. So it really was this kind of computer discovery that allowed that. Now, that said, so as we were then engineering the process so that we could capture these states, the toxic oligomers, the protofibrils, the plaques, we were characterizing their structures and their toxicities. Plaques aren't toxic. They're almost a dumping ground, a way to kind of prevent the toxicity. And so we showed that early on, you form these toxic oligomers, and then you get bigger aggregates. They become beta structure and such. And we were able to hone in on the toxic oligomers and ensure that our compounds are hitting just that. And so you don't want to take out the starting conformation of A beta 42, which is this intrinsically disordered signaling peptide that has function. You don't want to mess with that. And there's no need to really go after the plaques. Now, there's a little twist to that is if you have plaques, there can be a secondary nucleation mechanism to give you toxic oligomers. There's always back reactions on things. But if you can really get it earlier, it's not that the amyloid hypothesis is wrong. What I do is recast it and say it's the toxic oligomers earlier in the process that are setting off this cascade of events.
If you like listening to The Long Run, you'll love a subscription to Timmerman Report. This is where you can read my coverage of the most interesting startups in biotech, my weekly Front Points column, and commentary from a rotating cast of contributing writers. Individual subscriptions are available on a monthly, quarterly, or annual basis. Group subscriptions are available at a discount. Go to timmermanreport.com and click on subscribe for more. Okay, maybe you can talk here about the killer experiment that we talked about last year, where you did the retrospective analysis of the samples of people with Alzheimer's to really test, okay, how important are these toxic oligomers in downstream disease pathology?
Yeah, so we were, as a part of really characterizing the process and the structure of amyloidogenesis, we took one of our compounds that we had designed and we immobilized it to create an ELISA-like assay, a plate-based assay. And we were using it as a way, an indirect measure of alpha sheet structure so that we could move along this process and then detect the alpha sheet from other structures and then make a connection to toxicity. So we take those same samples, put them on cells and see if they killed the cells. So very simple, made that connection and showed a very high correlation between a hit with our assay and toxicity, which kind of goes back to that original hypothesis I mentioned. So then we were doing that on in vitro preps where it was clean and we could say, okay, we've got this conformer, we've got this one, we've got this one, this aggregate state, which ones show up in our assay? And all of that looked, so that was maybe not the killer experiment from the disease perspective, but to get us there, to really make those connections. And then we started collaborating with clinicians in the Alzheimer's Disease Research Center at UW, and they began giving us at first CSF samples. So we tested.
Cerebral spinal fluid.
Yes, sorry. And then we started testing that in our assay, and we were getting good results, hits. We did this blindly, and then we would deblind with them. And then we moved to plasma because they had matched CSF and plasma samples. And we were able to detect the toxic oligomers in plasma that was banked in some cases 20 years.
So these were samples from people who had Alzheimer's. We knew that, and that there was presence of these oligomers in both the cerebral spinal fluid, which is the most accessible fluid to the brain, and the blood.
Yes, that's absolutely the case. And so what we would do is we had cross-sectional and longitudinal samples, and that included controls that were not cognitively impaired. Now, this is according to clinical diagnosis, mild cognitive impairment, and then moderate to severe Alzheimer's. And so we would get these different samples, run them, and then just see, do we get a hit with our soluble oligomer binding assay, SOBA for Alzheimer's, SOBA-AD, or not? And found got very good results. We found that normal, non-cognitively impaired individuals didn't have toxic oligomers in their CSF or their plasma. And of course, if you can see this in plasma, that's preferable to a lumbar puncture. And then we did have to do some changes in the assay, develop the technology to be able to go down to such low concentrations, but we were able to do that. And then we got very high correlations between the AD and those with Alzheimer's disease at whatever level, from mild to severe, giving very strong signals in our assay. And then what was really interesting is we had 13 of what looked like false positives to us. And these were in the control group, cognitively intact. And it turned out that when we deblinded, some of them were cases that were known to convert and move on to mild cognitive impairment. So we were able to show that our assay was picking this up before the advent of cognitive deficits and before the conventional biomarkers, for the most part, could pick this up.
Wow, they weren't false positives at all.
They weren't false positives. And then we went further because that was only roughly half of those cases. And then we went into, got autopsy records and follow-on clinical records for 12 of the 13 what appeared to be false positive samples, and 12 of the 13, in fact, progressed to mild cognitive impairment. And then we didn't have any follow-up on the 13th.
Yeah, so this is how you end up. Your total pool, as I remember from the PNAS paper, of samples that you analyzed was 379.
Yes.
And what you found was that your peptide was 99% sensitive and 99% specific at detecting this.
That's correct.
Highly, highly accurate, and at an early stage, even before the presentation of symptoms.
Yes, that's correct. And then we also, it's not just Alzheimer's that can pick up, because remember I said these amyloidogenic proteins all of them we've looked at are forming the structure. So then we also looked at some Parkinson's cases. And these were not blinded. These were individuals getting surgery for deep brain stimulation devices. So they had confirmed Parkinson's, and we got their CSF, and they tested positive in our assay as well. So not for Alzheimer's, but for Parkinson's, because we can change out how we do the assay to be able to distinguish what the chemical entity is that our peptide is pulled down.
So you found an alpha sheet on its alpha-synuclein, right?
Correct.
Well-known target for Parkinson's.
Yes.
So wow, this is getting really exciting now. I think a lot of people listening, entrepreneurs and investors, are thinking, okay, there's some potential here for early diagnosis. We can talk about treatment later. But now let's come to this question about the company. Okay. When did this become because you had never started a company before. What made you think that this is something that needs to happen in a bigger way with private industry outside of my lab?
So a few answers to that, a few things that kind of came together. One, I've been supported pretty much continuously by NIH my entire career, as well as private foundations, Department of Defense, others. And you always you're writing grants. You wait a long time. You rarely get them the first pass. You revise, you go back in, and it's just a very long process. And it's for, great as it is, it's never enough money to do what you want to do. And you're trying to scrimp and save and get as much as you can out of this. And it's kind of grueling, and it takes a lot of time, and it limits often what you can do. And so in terms of going to your question about, why do it through a company, that's kind of the reason. It was like, okay, it's time to really make this happen. It would be a slow death, in my opinion, to go the SBIR route, the whole grant route, when the Alzheimer's field was heating up. It's something, I'm not getting any younger. I feel a duty because I've been taking taxpayers' money for a long time, and it's time to give back. We realized we had something that could really be a game changer, and it was just time to get it in the private sector. I've seen too many people try to do it slowly or within academia, and I don't believe it can be done. It's very difficult to do it that way. You have to get out.
And so you felt this personally,
Yes.
that you, now there are lots of ways to be involved as an academic founder, and they don't always involve you like leaving to become a CEO, like operating on a day-to-day fashion. Why did you decide to do that?
Well, it's still going on. So I still have my UW lab. And what's nice about it is that we can do basic research there. We can investigate different diseases and essentially do a lot of the early work. It still can be published, and it's great for students and such. And then we can develop the systems there. And then as we roll out different diseases, so the idea is we're not a one-trick pony. It's not just Alzheimer's. We've got an Alzheimer's program going, and we hope to get in the clinic this year with our therapeutic. Our diagnostic has FDA breakthrough status for Alzheimer's. And then we are also working in-house on Parkinson's, both therapeutics and diagnostics.
Okay. Well, that's at AltPep. That's so you're really wearing two hats.
I'm wearing two hats.
You have the faculty job is a part of your day job, and the other half is here at AltPep,
Right.
the company. But let's talk about the vision for this company, because you talked when we spoke last time about a complete solution.
Yeah.
And I think this is part of what really captivated me, and I think some of your investors who I know as well, the idea that you can detect Alzheimer's early, before any symptoms are apparent, from a blood test with a highly sensitive molecular test. Not asking a 40-year-old, did you lose your keys yesterday or something, like, and and making inferences,
Right.
which has plagued the field of neuroscience in many respects. So you start there with early diagnostic that has high predictive value, and then you've also, I don't think you mentioned this, but the ability of the peptide to capture the toxic oligomers. So like soak it up in a way and get rid of it before it accumulates and ends up in all these plaques and tangles and causes all those downstream pathologies. So you really have, as you say, a complete solution that's diagnostic and therapeutic, hand in glove. That's what you're driving toward here at AltPep.
Yes, that's absolutely the case. And so it's fundamentally, it's very simple. We have these compounds that are complementary to the toxic oligomers. They bind them. And when they bind them and they're on our assay, we detect, and then that same species is detected by, the peptide, bound by the peptide. And then when not attached to a plate, it stimulates the turnover of the toxic oligomers. And so what we find is we get very tight binding between our compounds, our alpha sheet designs, and the toxic oligomers. They then stimulate the microglia to clear the toxic oligomers.
Okay. So, but there's a lot of challenges in this field. Regulatory being one. Just the practicality of running such a clinical trial. Like if you were to enroll, otherwise healthy 40-year-old people, you can't wait around 25 years to see if they eventually develop signs of Alzheimer's to really, really confirm this hypothesis. So what's happened to make this a more practical and tractable sort of problem for AltPep to even exist?
Right. Well, we clearly can't wait 20 years to see the effect. And so what we're planning, so the idea is if we can get in early, I would hope we can just essentially prevent it from happening with therapy. I mean, it's not going to be one and done, but with therapy, and then keep the toxic oligomers levels low so that we kind of close the valve on all these downstream events. And then, but we have to get in, and most of the patients and people we would be looking at already have some symptoms. And so the idea for a clinical study is to get into cases in patients that have mild cognitive impairment, the early stages where we should see some effect. We should be able to stall the process, but we won't necessarily be able to make up for damage that has already been done. Now, where our therapy probably won't be very effective is if people have very severe forms of AD, because there's just, we can't turn back.
If you're 85 years old and your brain is full of these plaques, it's just too much damage has been done.
Right.
And you can't regenerate neurons and things like that. But if you're talking about younger people who have signs of mild cognitive impairment, you can design a study now to show that, on that molecular basis, you are clearing out those toxic oligomers, and hopefully you can follow them for 6, 9, 12 months to show that, gosh, we're seeing some difference in their clinical symptoms of disease.
Right. So I don't know in what sort of time period where we would see the cognitive benefits or the change relative to the non-treated groups. But now the door has been open to using biomarkers as well. So this is where our assay also comes in, is we can measure progression and therapeutic efficacy. We do this in our preclinical studies with our animal studies by using our SOBA assay. And then what we've got is essentially we see the drop in the tumor burden, in the toxic oligomer burden.
Now, that you said the door's open. That's from the Food and Drug Administration. Yeah. The FDA's neuroscience division has shown a willingness to use some of these biomarker-based approaches to measure progress against Alzheimer's, recognizing this very problem that we've been discussing, that it's just not practical to run 20-year clinical trials. There were a couple of events, I think prior or in the early days of the founding of AltPep, where the FDA approved a couple of cerebral spinal fluid molecular diagnostics, the first of their kind, to detect Alzheimer's.
Right.
What did that say to you, and how did that alter your strategy at the company?
So yeah, and it wasn't that long ago. So you're talking about, I think, Fuji Rebio and Roche, and they both are CSF tests, as you mentioned, and they both involve A beta 42, which is our target, is the toxic oligomer. But they use it different ways, and they measure different things. But that said, it's still that target in essence. And so that affected, I think, when we turned in our paperwork to the FDA, we were initially proposing kind of steps, which was to develop an exclusionary test where we would just exclude Alzheimer's based on our assay. And then we would go for affirmatory, thinking, okay, we got to socialize this. This hasn't been done before. We're going to work our way up. And then, of course, I'll say the moonshot is we want to screen everybody 40 years of age and up, almost like a cholesterol test or something, or PSA, and just really get in there and know when people are doing this early. But that, of course, is in the distant future. But so then you have this case where we were going for this approach. The FDA came back and rewrote our intended use, and they gave us more than we asked for.
Wow.
Yeah.
This doesn't usually happen.
No, it was my first time. I didn't know, but yes, we found out, and that doesn't usually happen. And so they basically took us right up to, skipping that first exclusionary test. And at the time, it didn't really make sense, but they rewrote it. We said, okay, fine, that looks good. Well, we're skipping a step, and it's part of a workup, a larger workup to make this diagnosis. Then when Fujifilm Bio got their approval, it was like there was our intended use. And then when Roche, and basically they set us up, and we had been going in as a de novo device. And so that allows us to use the other two as predicate devices. So it really helps our case, in fact. And in another way, it helped because they used particular samples and where PET imaging had been done as well on these individuals for validation. And so we know the samples that they used, and we've got them in the freezer here. So we're going to be using them for our validation.
So this should speed up and reduce the cost to get the kind of answers you need to in some sort of robust, both retrospective and maybe even prospective fashion.
We probably won't have to do, the FDA hasn't required prospective studies of the other two. And that was really good news because we were assuming we'd have to do a prospective study.
Mm-hmm.
And so it doesn't look like we will have to, especially with predicates and the same samples and that sort of thing.
Well, and with the really robust data set like you described from the retrospective studies that we do now have the benefit of time of seeing that they did eventually manifest clinical symptoms.
That's right.
Okay. So now therapeutics, can you talk about are you using the same agent, the same peptide that does this binding, has this binding action very specifically, or are there some important differences?
So it's the same in the sense that it's an alpha sheet peptide. It's part of our design template, and it works from there. But it is a different sequence and some slight changes as well from there. And so essentially the same but different. And you have different properties you want for something that we're delivering intranasally than something on a plate, so to speak. And so. So in this way, since we were able to patent the structure, we can make modifications to the sequence to tailor it for specific binding to different disease cases, the different amyloid peptides or proteins involved in those, but also for different applications. So, for example, the peptide we use for the plate assay is a little too hydrophobic and sticky for a therapeutic. And so, considerations, just physical chemical considerations like that. And so it's a different peptide, same class. And then it works the same principle, the binding and then preventing further aggregation.
But you build off of the same template.
Correct.
It has that same binding, those characteristics, the affinity, all of that, the ability to capture those toxic oligomers. And you're going to engineer in therapeutic properties, say the intranasal delivery and the right kind of half-life that you want.
Yes.
But back to this idea of the complete solution, in theory, like someday these products are on the market, that they could work together as companion a diagnostic and therapeutic 1-2 punch.
Yes.
If you detect certain amounts that we think are, trending in the wrong direction for a 40-year-old or a 45-year-old, nip it in the bud before it even becomes a problem.
No, absolutely. And so we early on demonstrated too that the same peptide that we use on our assay is an inhibitor. It just doesn't have, we could improve upon its solubility and different features. And so it really is the same thing going into this is kind of, the 1-2.
Mm-hmm. Mm-hmm. Okay, so now you back to one of those hats as the entrepreneur, CEO. You're out raising money, talking to prospective partners, I suppose, hiring people. How is this? Does this suit you?
Are you questioning that?
No. How does it feel? Because you definitely didn't have to do this. You could have stayed on the faculty forever, and a lot of people do, and that's just fine. And get somebody else, maybe from the outside business world, to run the company.
Right.
But no, you seem to be enjoying this.
I'm really enjoying it. I didn't know I would. in academia, you get your own funding, you hire people, you do all these things. It's like you're a little mini company. And so it's clearly different in biotech, but you do learn some of those skills along the way. You're juggling all these things, but it goes back to that, kind of feeling a duty and feeling like this is the time. This is something that could really be important, and there's nobody that knows it better. And I want to see this through. And I'm not doing it alone. I've got a great team, and I have enjoyed it. It was a little rough at first with fundraising and pitching and such, but you find the right one thing that I didn't really appreciate before. I hadn't had contacts with venture capitalists and such. Is you get the right venture capitalists, they've got their scientists, they've got great analysts. You're basically talking with scientists about your science, which is what you do in academia, and then come other things. But I actually enjoy it.
Who has encouraged you or supported you on this journey?
Well, many, I'd say. Initially getting going, my husband, and he had been an entrepreneur and had companies, tech companies and such, and saying, you can do this, you can do this.
Uh-huh.
And so, and then my department has been very supportive, the bioengineering department, Commotion at UW in this valley of death time with money, with introductions, with all kinds of things. So there have been many over the course of this process. And then I'd say others, it's been starting the company. I'm going to put Chad Robbins in there because he's been extremely helpful. And then we've got some great investors that are involved, and it's there have been so many. I probably should get the list going so I wouldn't forget anybody. But there really have been a lot of people who have been extremely helpful along the way.
What are the next big steps on your agenda here for this next year or the next couple of years? What are the big goals?
So, want to get the FDA approval for our assay, for our Alzheimer's assay, bring the Parkinson's along after that. We want to get into the clinic, get into humans with our Alzheimer's therapeutic, which we call Alt100. And we're in the process there, expecting GMP material for that in March. We're working on the GLP toxicology, all the things you need to do, the IND enabling work. We had our first pre-IND meeting with the FDA to, vet our plan, that sort of thing. So I'm really excited to get into humans because we've got really, I think, very encouraging preclinical in vivo studies in animals. But you just, of course, you never know until you get into humans. And so I'm very anxious to do that.
And you mentioned that Parkinson's program. That's a little bit further behind in the pipeline?
By design. So what we do is we bring in people that are, some of them have experience in neuroscience, some don't, some are more analytical, all kinds of different areas. And they learn about alpha sheets and learn about these systems. And then the idea is, as we move into the clinic with some, rather than like some companies do, you have to lay off your R&D people, is keep them because they've got this great expertise now with these systems and then apply them to new diseases. And so that's already happening. And they are doing animal studies for Parkinson's and such in the early preclinical studies.
And the same conceptual framework applies. Catch the alpha-synuclein early before it manifests with tremors and these other symptoms of Parkinson's, and then come in with a very similar peptide that binds with it and clears it.
Yes.
Before it accumulates and it's too late.
Exactly. And then we've got a couple other programs waiting in the wings as we move through with Alzheimer's and Parkinson's.
What's one?
Which is why the link to UW is important, because we establish these systems, and then can bring them into AltPep as we move along. So we're not going to stop. The plan is to go after many of these diseases.
Yeah. And so you have the systems, the processes, the collaborations still in place with University of Washington biology colleagues, right, as well as access to clinical partners.
Exactly.
In the medical school. And now at the company, you've been able to extend your network of valuable helpers, so to speak, with an investor syndicate that provides some capital to execute on these plans, but also some advice, some recommendations. Folks like, I know Mike Polini of Section 32, former CEO of Foundation Medicine, knows a thing or two about the diagnostics business.
He knows a thing or two about lots of things.
Eli Lilly is in your syndicate.
Yes.
They've got a lot of experience in Alzheimer's disease.
Yes.
A lot of relationships there. What's one surprising thing about, your new life as an entrepreneur? One thing that you never would have guessed 5 or 10 years ago?
Oh, it's kind of a hard question. I guess, well, the thing that's coming to mind at the moment is kind of relates to what we were talking about earlier is it okay, it sounds trite, but it takes a village. And in academia, even though you have colleagues and collaborators, you are, and you have a department, it's not that you don't have help, but you are responsible. You're siloed in essence. You're supposed to get your funding, you do your hiring, you do your management, you do, all these different things. And it's not that you can't get advice, but you're doing it. You're the principal investigator and you're responsible for it. And getting to AltPep, I'm not the only one. I've truly got a team. I've truly got, and it's a big one. It's everybody out there. And it's fun. It's fun to work with people. It's fun to have those connections and debate things and, plans and how to move forward and to really have people you trust and you're doing it together.
Mm-hmm. Mm-hmm. And it's not the dark side.
No.
It's a continuum, actually,
right.
From basic through applied research.
Yes.
People helping each other,
Yes.
work with their eye on the prize, that big problem that our society has. And I know a lot of people listening to this must be really rooting for you and hopeful that this works.
Well, we're going to give it our best shot. I mean, we're very hopeful, and we're not going to stop until we see how this works in humans and push these along. And there are just too many people afflicted by these diseases, and the head count isn't really known given the lack of good diagnostics and because of these long lag periods before people show symptoms. So
it's tens of millions of people.
Yeah.
It's just in the United States.
Well, amyloid diseases in general, it's estimated 1 to 2 billion people.
Yeah. And
that's, again, without diagnostics for most of these.
And if you could do this with a blood-based diagnostic that people get a checkup when they're in middle age, that has really big potential for human health.
Yes. Oh, it's like a dream come true. I mean, to discover something in the computer, think it's an artifact, and then chase it down and maybe, I'm ahead of myself here, but this is a dream come true to be able to do this.
It's the culmination of 30 years of work.
Yes, it is.
Well, I think that really epitomizes the long run. Valerie Daggett, thank you so much for joining me today on the show.
Thank you very much, Luke.
Thanks for listening to The Long Run, a production of Timmerman Report. Pedro Rosado of Headstepper Media was the sound editor. Music is from D.A. Wallach. See you next episode.